Hey everybody, this is Brittany. Welcome back to my channel. This is going to be the first of a 3-video series on GMOs, or genetically modified organisms. Being part of the gardening community, I'm well aware of how controversial a subject this is, but I really wanted to give my 2 cents on it because I did complete a Bachelor of Science at the University of Guelph, and as a result I took a lot of classes on molecular biology, cell biology, and so I do feel like I know something about how genetically modified organisms come about. I think a lot of the fear and the nervousness surrounding GMOs is really built on a lack of understanding of sort of the core processes of DNA and how GMOs actually work and how the inserted gene interacts with, say, the already existing genes. Now, just to be clear, I definitely think that there are pros and cons to GMOs. I'm not 100% for or against. I think, there is more nuance to it than that. So the next 2 videos are really going to be about debunking myths and kind of my final thoughts about GMOs. So this video really serves the purpose of providing basic information, background information, for those videos. Now, I think we're all reasonably familiar with DNA. We know that it's sort of the genetic instruction manual of our bodies. We've seen the kind of spiral ladder form that it takes. As you can see in that picture, there's kind of the sides of the ladder, and then there's the rung, which actually has 2 parts.
And those 2 parts are the nucleic acids, which is what we'll talk about for the purposes of this video, because they're really the most important parts of the instruction manual. So basically the ladder is made up of 2 strands that can be kind of like zipped, unzipped away, and that's really what happens when it comes time for the body to form another protein, which is mostly what our bodies are made of proteins. And it's the DNA that tells the body what the protein is going to be constructed from, which are amino acids. They're always constructed from amino acids, but what amino acids? Because there are about 20 amino acids, and the different order and configuration of the amino acids, the sequence actually, that's probably a better word, the different sequences of the amino acids is ultimately what determines the function of the protein.
So there are 4 nucleic acids, we'll call them A, T, C, and G, and you can have quite a variety of different sequences. Say you have a gene that's maybe 90 nucleic acids long. Of course, you have a multitude of possibilities in terms of the sequences of the nucleic acids making up those strands.
So the 2 processes that make up the sort of interim between DNA and protein synthesis is called transcription and translation. Transcription is when a copy is taken of one of the strands of DNA, it's taken out of the nucleus to the ribosome, which is a cell organelle, basically just a piece of machinery that then translates that strand into a protein. And on one side of the ribosome, it reads the strand. On the other side, it puts together the strand of amino acids. And for every 3 nucleic acids that it reads on the strand, that's called a codon. And a codon is always associated with a particular amino acid. So it reads a particular 3 nucleic acids, and it knows, okay, we need this amino acid.
Now, when the codons are made up of 3 nucleic acids and there are 4 possible nucleic acids, of course that means that there is a variety of different codons that can be formed from that. So sometimes multiple different codons will lead to the same amino acids.
And it's also important to note that there are what we call start codons and stop codons, meaning that at the start of a gene, the 3 nucleic acids are usually the same because they code for an amino acid called methionine. I believe it's methionine that always starts off with protein. And then there's usually the stop codons, which I think there's a couple possibilities of combinations of nucleic acids that basically tells the ribosome, okay, stop making this protein.
So it refers to an organism that comes about as a result of scientists taking a gene from another organism, something that they want to see in a particular organism, and inserting it into the genome of that organism. Of course, the genome being the DNA. And the whole purpose is because they want a particular organism to produce a protein that it wouldn't otherwise produce.
What's important to note about that is that once that gene is in the genome, the only ever time it really kind of interacts with anything is when it's going to be transcribed and translated. Everything that's happening to it is completely independent from anything else that's happening to any other gene. So once it comes time for another gene to be translated, a copy is made, it's brought out to a ribosome, and these two genes never interact. These two genes have no possible way to have any influence over the translation of proteins that has to do with one or the other.
Thank you very much for watching, and once I film my other 2 videos, I'll post them up there, and I hope you'll be interested in watching them. I know that some of you might be from the school of thought of, GMOs are horrible, and some of you might just not know what GMOs are, and I really thank you for having an open mind about this, and I hope that you'll come with me on the rest of this journey.